Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Rabbi zidni ilma. This is going to be the next part of the previous lesson that we started about the cell cycle, and in which we discussed about the interphase, which was actually the preparatory phase of the cell before it enters into the cell division properly. So just for a recall purpose, we talked about the cell cycle. It is actually the cycle, or it is actually the phase between one cell division to the next cell division. So when there is one parental cell and it is going to be divided into two daughter cells, and when the newly formed daughter cells get mature enough to start their own cell division, then the next, from first cell division till the next start of next cell division, is actually a complete period, which is called as cell cycle. So cell cycle, when we talk about, was the first phase that was called as interphase, and now we are moving towards the M phase of the cell cycle, which is actually the reproductive phase, in which two cells are formed which are identically same as the parental cell, as with special regard to the chromosomal number. Now, first of all, before going into the detail of mitosis, we should know what is mitosis actually. It is the division of nucleus into two parts called as karyokinesis, which is succeeded by the division of cytoplasm, and finally two complete new cells are formed. So division of nucleus into two parts, also called as karyokinesis, and it occurs only in eukaryotes. Recalling from the previous lesson, I have already mentioned that in case of prokaryotes, we do not have the process of mitosis, rather we talk about the binary fission. While in case of eukaryotic cells, there's a proper type of reproduction in which cells divide following multiple phases, multiple stages, multiple steps, and these steps are actually called as mitotic phase or mitosis. It has total 4 stages, and it doesn't occur in some cells such as the brain cells. And why doesn't it occur? That there are certain type of cells in our body which just stop dividing once they are formed. For example, your nerve cells. Nerve cells, once made, can never ever again divide to make new types. So that's why the injury to the nerve cell is irreparable. So mitosis occurs in those cells which get damaged, or which need to be replaced by the new cells, or which are just growing in their number. So if we talk about specifically about the nerve cells or the neurons, these are something which are once formed will never be repaired or will never be grown again. So that's why mitosis is not the part of the cell division cycle of the nerve cell or brain cells type. So that is the major reason behind the aging, that aging cannot be stopped. Then what are centrosomes? These are certain structures which we earlier discussed about these cell organelles when we were discussing in the chapter 4, that there are certain cell organelles which are responsible for different specific functions, and we talked about the centrosomes there. A centrosome is an organelle produced by the cell in the G2 phase of the cell cycle, and it helps in the mitosis. How? Because two centrosomes are produced or present in the cell, and during cell division, each of the each centrosome has two centrioles actually, and each centriole moves to the opposite end of the cell just to pull the chromosomes apart, and for the purpose of division of the cell exactly into two halves. So centrosomes are actually the present in the cell, which is consisting of two centrioles each. And centrioles, what is the main function behind the centriole? That is the pulling apart of the chromosomes during cell division, so that nucleus can be divided and cell can be finally divided into two daughter cells. So that is the position and purpose of the centrosome. Here we can see that centrosome has two centrioles, and each centriole is actually having 9 triplets of microtubules. 9 triplets means that is in total 27 microtubules in one centriole, and there are 2 centrioles in one single centrosome. Now specifically moving towards the stages of mitosis, the first one is called as prophase. As the name indicates, prophase is the first or the initial phase of the mitosis. Pro means first, phase means stage. So it is the first phase of mitosis. The chromatin present in the nucleus condenses to form chromosomes in this phase. Again, if we recall from the previous lecture, I have already mentioned there that the difference between chromatin and the chromosome is the chromatin is actually the thin and fine fiber-like mesh network-like material which is present in the nucleus, while when the cell is going to be divided or it is going to enter into the cell division, the chromatin is first converted into chromosome, which is actually thicker and condensed as compared to the chromatin, and chromosomes are visible while the chromatin was invisible. And these chromosomes, when these are visible, are actually going to be because of the condensation process that these pass through. And this condensation process is actually the part of prophase. Then the second thing, the centrosomes begin to move apart from each other, and that's what we have already talked about, that the centrioles and centrosomes are going to the opposite ends for the purpose of division. Later, these centrosomes also start the formation of spindle fibers or microtubules. And if we again recall from the previous lecture, we talked about the G2 phase in which spindle fiber proteins were being made, and these spindle fiber proteins which are going to be made are actually the stretching proteins or the stretching fibers which will be connected to the chromosomes to pull the parts apart for the purpose of cell division. Here you can see that we have a nucleus in which the nuclear envelope is actually going to be broken down. The centromeres are there and the chromatids are attached to the centromere and the kinetochore. And there are centrosomes which were actually together, but now they are going away from each other to the opposite poles. And thin, fine fiber-like structures are going to be produced out of the centrosomes or the centrioles, called as microtubules. And overall, this centrosome, along with the microtubules, is collectively called as mitotic spindle. And these microtubules will give rise to the spindle fibers, which will later be attached to the kinetochore of the centromere of the chromosomes for the pulling apart condition. Next phase is called as metaphase. As the name indicates, it is meta means middle, phase means stage. So it is the longest stage of the mitosis, lasting about 20 minutes. Centrosomes align at opposite ends of the cells. Chromosomes get arranged on an imaginary metaphase plate. So metaphase plate is actually the equatorial line or equatorial plate, which is exactly at the center of the cell, at the central point of the cell, and which is making an equator, and the cells chromosomes are actually aligned into the exact half of the cell. The chromosomes are attached to the spindle fibers with the help of kinetochores present on centromere of chromosomes. Here you can see it, that these chromosomes are actually aligned just at the center, and there's a kinetochore protein on each side of the centromere, and these kinetochore proteins are responsible for the attachment of spindle fiber with the centromere. So centromere is not directly attached to spindle fiber, rather centromere is attached with the spindle fiber through kinetochore proteins. So this centralized chromosome position is actually called as metaphase plate. So spindle poles are formed. Centrosome has originated its spindle fibers, which have been connected to the chromosomes at the kinetochore point. Attachment has been done. Chromosomes have been aligned exactly into the center, and in the next phase, which is called as Anaphase, which is the shortest stage of the mitosis and lasts for just for a few minutes, these two sister chromatids of each chromosomes, which are actually connected at the centromere position, now will be pulled apart by the pulling spindle fibers. As we have previously mentioned, it has a stretching ability. It has ability to stretch to the opposite sides, so the sister chromatids will be pulled apart. Each chromatid behaves like an individual chromosome. By the end of Anaphase, the two ends of the cells have equal number of chromosomes. So every single chromosome is now have moved to the opposite poles, and here we can see that we have got two different distinctive bodies on each poles, which will now be considered as two different nuclei after the telophase. So microtubules or spindle fibers are there. Shortening of kinetochore microtubules has occurred, which has pulled actually the chromosomes towards the opposite poles, and the increasing separation of spindle poles actually has been observed. That means elongation of the cell. Finally, the last stage, which is the telophase, the nucleus develops surrounding the chromosomes at each end of the cell. The chromosomes become less condensed. That means the chromosomes is now going back to the chromatin network point, because the condensation that was actually made for the purpose of formation of chromosome has been achieved. So chromosomes will now be back converted into the chromatin network. A nuclear envelope which was broken down earlier will now begin to develop again to form around each nucleus. So mitosis is thus completed, and here you can see the reforming of nuclear envelope is occurring, and the things that are perfectly designed into two distinct nuclei. Finally, when the nucleus have been completely divided, now there's a step of the cell cytoplasmic division, in which the cytoplasmic contents will also be divided to make the cell division complete. Cleavage of cell into two halves is going to be occur. Each half is called as daughter cell. Each daughter cell has the same number of chromosomes present in the parental cell. Here you can see the lengthened cell is now going to be pinched inside, or pinched from the center, and the groove will be formed, and this groove will continue to meet the other end till two cells are completely divided. So these chromosomes will be decondensed back into the chromatin material, the spindle fiber will be disappearing, the nuclear envelope will be completely formed, and finally we have got two different cells which will now be able to start a new interphase for the purpose of new cell division. So another interphase is the stage after cytokinesis, right up to the next prophase of the mitotic cycle. It again consists of G1S and G2 phase of cell cycle. It ends with the beginning of prophase of the M phase of the cell cycle. Hence, it can be said that two successive mitotic phases are separated by interphase. And finally, the cell which has just been formed is back into its perfect shape. The nucleolus have been reappeared, the nuclear membrane has reappeared, the chromosomes are back converted into chromatin, and the two centrosomes are clearly visible on one single pole. Now this cell will start again its interphase, and whole cycle will be repeated. So that was all for the mitosis and its 4 stages, and thank you so much. Jazakallah khair. In next class, inshallah, we will be discussing about the meiosis, or the second type of division, which is actually responsible for the purpose of gamete formation.